Hello and welcome to BioCyc. This webinar will introduce you to the information present in BioCyc and to the many tools available to help you search and analyze information in BioCyc. Our overall mission at BioCyc is to provide you with up-to-date and comprehensive information on microbial genomes and metabolic pathways, together with powerful bioinformatics tools for searching, visualizing, and analyzing that information. This first webinar in the series will give you an overview of the functionality present in BioCyc. BioCyc has several main use cases. Overall, it's a web portal that integrates curated data for many genomes with powerful bioinformatics tools. First use case is that BioCyc is an encyclopedic reference on sequenced genomes and pathways where you can look up information on genes, metabolic pathways, and more. There are 56 curated databases listed at the URL here, and overall the contents of BioCyc are derived from curation of 95,000 publications. There are 17,000 total databases in BioCyc, most of which are microbial, and BioCyc integrates a broad array of data, including metabolic pathways, regulation, and gene essentiality data. It has a number of sequence analysis tools. That's another of its use cases. A third use case is transcriptomics data analysis, as well as metabolomics data analysis and comparative analysis at both the genome and the pathway levels. Here's the overall process that we use to create new pathway genome databases, as we call the BioCyc databases. It combines import of information from a variety of databases with computational inferences. The process begins with an annotated genome, most of which are obtained from the NIH RefSeq database. The computational inferences we apply predict metabolic reactions and transport reactions of the organism. They predict metabolic pathways as well as the genes that fill holes in those metabolic pathways. Our algorithms predict operons, compute orthologs, and compute PFAM domains. We then import regulatory data from RegTransBase, subcellular location data from pSortDB, GoTerms and protein features from UniProt. We also create database links to a variety of external databases, and we include organism phenotype data and gene essentiality data. All BioCyc databases contain these computational inferences and contain some imported data depending on which data are available for a given genome. That gives us a basic Tier 3 database, as we call it. Then, for selected databases, we'll apply curation to yield either a Tier 1 highly curated database or a Tier 2 moderately curated database. Among the tasks our curators perform are importing additional protein functions from UniProt, performing literature searches to find new gene functions and pathways that are entered into the database, authoring mini-review summaries of genes and pathways, updating a variety of database fields such as gene names and synonyms and activators and inhibitors of enzymes, entering new enzyme reaction associations, repredicting metabolic pathways and reviewing the predicted pathways, reviewing and extending transporter and transport reaction associations, and defining regulatory sites and interactions. Our most highly curated database within BioCyc is the EcoCyc E. coli database, which has been derived from the curation of 38,000 publications, and it contains a wide variety of information, from gene ontology annotations to protein features to multimeric complexes to regulation of gene expression. Now, in the encyclopedic reference use case, BioCyc provides extensive search tools to help scientists find the information they're looking for and extensive visualization tools to speed the assimilation of the information that's found through these searches. BioCyc provides a variety of genome informatics tools, including gene, protein, and RNA search tools and genome site search tools. So the user can search for genes according to this large number of fields. There's a gene information page that lets the user retrieve the nucleotide sequence of a gene or genomic region or the amino acid sequence of the gene product. There's a transcription unit page that shows regulatory sites and interactions. There are blast search, sequence pattern search, and sequence alignment tools. There's a tool that will map SNPs to the nearest genes and show effects on translation. And there's a genome browser that can zoom from the full chromosome in one screen to the sequence level. It has a comparative mode and a tracks facility. It enables omics data painting, and it can produce a genome poster. Now here is our search tool for genes applied in this case to a Salmonella database in BioCyc. And essentially it lets the user specify any combination of these different fields to search by the protein, the name of the gene or protein, its sequence length, its map position on the chromosome, which replicon we want to search, as well as these other searches such as searching by PI or molecular weight or small molecule ligand. And here's an example result page, in this case for a curated Salmonella transporter, that shows a mini review summary authored by our curators. It shows the subunit structure of this complex. Here's a B subtilis curated protein, in this case a transcriptional regulator, so the Regulon tab shows the operons that are regulated by this regulator. This is our genome browser in its comparative mode, which shows 6 different genomes aligned around this orthologous gene here to see conserved and non-conserved regions. BioCyc also provides a variety of pathway informatics tools. There's a pathway search and a pathway information page. One can customize pathway diagrams for publication and create personalized multi-pathway diagrams. There are comparative pathway analysis tools. There's also a reaction search tool and a reaction information page. The reaction pages show reaction atom mappings present in our databases. There's a metabolite search and metabolite information page. There's also a zoomable organism-specific metabolic map diagram for each organism in BioCyc, from which a metabolic map poster can be generated. There's a metabolic route search tool that computes optimal paths from a starting metabolite to an ending metabolite, and it's possible to execute quantitative metabolic models through BioCyc. Here's an example pathway page, and in this case for Synechococcus elongatus, along with a pathway mini review from the Metacyc database. And here's a metabolite page for arginine, again in Synechococcus elongatus, that shows all the reactions in this database involving arginine, both that consume and produce the compound. Let me now introduce you to several BioCyc tools that will be described more fully in later webinars in the series. These tools are useful both for navigating the genome metabolic regulatory information space and for analyzing high-throughput data sets. Our first tool is called the Cellular Overview Diagram. It's an organism-specific metabolic map diagram that is zoomable and searchable. Each node in the diagram is a metabolite, and we can identify them by mousing over them, and each line is a metabolic reaction. The diagram can be searched using the commands on the right side, and it can be zoomed using the mouse scroll wheel. And we see more information as we zoom in, and we can drag the diagram around to position it. You can also compare the metabolic networks of two organisms by highlighting on this diagram the reactions shared with another organism. BioCyc has a number of omics data analysis capabilities for transcriptomics, proteomics, and metabolomics data. The user can paint these large-scale data sets onto individual pathways, multi-pathway diagrams called pathway collages, and onto the full metabolic map cellular overview that we just saw. There's also a tool called the Omics Dashboard, and there are standard enrichment analysis tools to compute pathways that are statistically overrepresented in gene or metabolite sets. It will also compute overrepresented GO terms and overrepresented regulators. This diagram shows gene expression data painted onto a single pathway. We can also paint omics data onto the cellular overview diagram. In this case, we're painting a transcriptomics data set onto the E. coli cellular overview diagram. And you may notice that This is an animated display in which we're watching the progression across 6 different time points during the shift from anaerobic to aerobic growth. And we can stop and start the animation, and we can display the expression levels of any gene or genes within the data set. The next tool is called the Omics dashboard. The Omics dashboard provides an integrated view of a metabolomics or transcriptomics data set. In this case, we're looking at the same gene expression data set for E. coli. The Omics dashboard consists of a set of panels for major subsystems like biosynthesis, degradation, and energy metabolism. And each panel contains a set of plots of the subsystems within that system. Within each plot, the 6 vertical bars correspond to the 6 time points, and the fat dots average all of the genes at each of the time points, so that we can see that overall during this shift from anaerobic to aerobic growth, amino acid biosynthesis is going up. Now, if we want to see more detail about one of those subsystems, we simply click on that plot, and we now get the same style of display for each of the individual amino acid biosynthetic pathways. And if we click on it, we see details of each gene involved in the pathway at each of the different time points. Another tool within BioCyc is called SmartTables, which allows the user to collect and save lists of database objects, such as lists of genes, metabolites, or pathways, or sequence regions. The user can explore properties of that set interactively, filter and combine these sets, such as by taking an intersection of 2 sets, transform them into related objects, like transforming a gene list into a pathway list, and sharing SmartTables with the public or with specific collaborators. For example, here's a gene SmartTable that was imported from a file, and we're looking at the name of the gene product, and we've added a transformation that shows for each gene the pathways that that gene is involved in. Here's another gene SmartTable, but in this case we're looking at the accession number for each gene, its start and end position within the genome, and the DNA sequence of the gene. Here's a SmartTable for metabolites, in which we're looking at metabolite name, SMILES string, and the structure of the metabolite. And in the next webinar in the series, you'll learn how to search BioCyc.